begin. So let's move on to the next part, part 4. And then we focus on CRISPR screens. So the CRISPR systems are originally adaptive immune system in bacteria that they use as a defense against viral genes. But scientists have modified it for genome editing. So the components of CRISPR system include mainly the guidance RNA, the sgRNA that recognize a specific position in the genome, and the Cas protein that which is an endonuclease that cleaves the sequence at that position. And we can adopt this system to perform gene knockout. So we design sgRNA or guidance RNA that targets a specific gene, and this Cas9 endonuclease will cleave that gene. So in this way, we can perform gene knockout. This is a brief overview of CRISPR screen. So we first construct a sgRNA guidance RNA library, which contains many different types of RNA targeting many different genes. And we transduce cells with the sgRNA library, usually through the use of viral vectors. And since sgRNA targets many different genes, they can perform genome-wide knockouts. And based on cell survival, we can see positive or negative selection of genes. So positive selection means that a knockout of a gene leads to cell survival. That is, cell expressing sgRNAs for these genes will be actually enriched. And we can use positive selection to find, for example, drug resistance mechanism. For example, after transduction, we apply to the cell culture a drug treatment or a drug treatment, and we will see if like under this drug treatment, certain genes with cells with certain genes be knocked out are preferentially enriched, and it will suggest that these genes may be related to drug resistance mechanism. Yeah. And for negative selection, inactive selection, knockout of genes will lead to cell death. So that means cells expressing sgRNAs for these genes will be actually depleted. And clearly, this can be used to find survival essential genes. Like if it is knocked out, the cell will die. This indicates that these genes may be crucial for cell survival. And in this part, you will be asked to analyze CRISPR screen data, focusing on positive selection and negative selection of genes with MAGIC, which is a powerful bioinformatic tool developed by Dr. Shirley Liu's lab. And for your convenience, we have already installed the MAGIC environment on Canon for you. So you need to first load Anaconda and source activate this MAGIC environment. And you can use this command MAGIC help to see help documentation for its command. And the CRISPR data is stored at this position. So remember that last time, I think accidentally deleted the master environment. And this time we make sure that these data files and the environment are locked to students, so students won't have writing permission or editing permission to these files. But when you run any code, do check that you don't write any of your results or change any part of the files in the original data file or the magic environment. So for question 4, it will at the start of part 4. At the start of part 4, it will ask you like to look at the QC metrics of magic, but really you need to first run magic and generate the files yourself. So we provide you the CRISPR screen data at day 0 and day 23, and you need to first generate the count table for sgRNA. So this is the command you will be using, magic count. You need to give it a library file that contains the sgRNA library information, including its ID, the sgRNA sequence, and the gene it is targeting. But we will provide this in the CRISPR data directory. You will also need to specify a prefix for your output file. Here I just use out, but you can choose anything you like. And you need to give it sample label indicating like what are the samples you have in your raw FASTQ files, and then you will provide a FASTQ file. Do note that for the FASTQ files, replicates under a same condition are separated by comma. So we actually have 4 FASTQ files provided, and the 2 replicates for day 0 and 2 replicates by day 23, and they are separated by a comma. But for samples under different conditions, like day 0 versus day 23, they should be separated by a space. So you will get a count.txt file as the output using this command. And here, this is an example I grabbed from the magic tutorial page. And it has columns of sgRNA, the gene it targets, and the sample names. And in the columns of each sample, it contains the count for the sgRNA. And to test if genes are positively or negatively selected across conditions, you should use this command, magic test, and give it the count txt file you got here, and then specify the direction of comparison. Here we like T is like treatment, C is like control. So we compare the CRISPR data from day 23 to day 0, and the prefix is specified using -n, and you can choose whatever you want. And do remember that the labels you use in the magic test command should match those where you are running the count command. That is, the labels here should be the same as the labels in the columns here. And you should consult this page to get an idea of what each file in the output means. And here is some sample script, bash script that you probably want to use to run this command, because these two combined, I think when I test it, lasts for 1 to 2 hours. So you don't want to wait it using the interactive command, but you should submit it as a bash script. And this is some recommended option parameters. And do remember to load Anaconda and the image environment first and put your code here. So finally, to question 1, we ask you to look at the QC metrics. So after running the above commands, you will get QC metrics stored in this count summary.txt file. And you should check this page for QC guidance. It contains information of what each column corresponds to which QC metric. And you will be asked to check whether the percentage of mapped reads is above 0.6, whether the zero count is less than 0.1, and whether the Gini index is less than 0.1. So we give you 4 samples, and if these are true for all samples and the data is of good quality. And another layer of QC metrics is ribosome genes. And ribosome genes are vital for survival. So knockout of ribosome genes will result in cell death. In other words, they are usually used as negative controls. So the gene summary.txt file contains genes ranked by how negatively they are selected. And since ribosomes are vital for survival, they should be ranked high as very strongly negatively selected genes. But the question is how to select rows corresponding to ribosome genes. A hint here I will provide is that ribosome genes start with RP in their names. So you might want to take a look at the documentation for this function grep and figure out how you subset strings with a certain pattern, like starts with RP. So you want to probably subset to row names that all starts with RP. Yeah, and the third layer of checking QC is to check replicate consistency. So we want to see that the replicates for day 0, for example, are quite consistent. The results we got are quite consistent. So here's a question for you to think about, and this is very vital for this part, is how do we count each replicate separately? A hint is that think of what I talk about here, that replicates are separated by comma. What if we separate each replicate by a space? So treat it just like they are samples under different conditions. And in this question, you will get a count normalized txt file, which contains 1 column for each of the 4 samples. And you want to check replicate consistency by plot replicate 1 versus replicate 2 and check their correlation. For example, for the data, the file here you check the correlation between replicate 1 and replicate 2 for day 0 and replicate 23. Replicate 1 and replicate 2 for day 23. And even easier approach is to plot the heat map of the correlation. And you want to see that the 2 replicates under the same day should be very highly correlated. And for question 2, we ask you to subset those positive and negatively selected genes and figure out what pathways they are enriched in. So in this time, you want to use this gene summary.txt file, and you should read it with a function here, read RA. And you will contain a column for score and another column for FDR. And you use score to determine the direction of selection and FDR below 0.05 to determine the significance. And after you get the positively and negative selected genes, you just run Bayesian or gene set enrichment analysis, just like you did in homework 2, to get enriched pathways. And then in question 3, we ask you to find the possible drug targets among the negatively selected genes. as you know, negatively selected genes are vital for glioblastoma cancer cell survival, so they can be potentially be uh, drug targets. But here's a problem. What if these genes are also negatively selected for normal cells? That is, to give you a simple example, if we knock out the gene that is essential for energy production in cells, we can definitely kill cancer cells, but it will also kill normal cells. So we really need to consider collateral damage uh, when we choose drug targets. One way to see if a gene is important for both cancer cells and normal cells is to visualize their expression versus dependency across many cell lines. So the more negative the dependency is, the more depleted it is in a gene is in CRISPR screen. So you will go to a website and check uh, for the potential drug targets, their expression versus dependency. So if you see that uh, its expression is highly correlated with dependency, which means that the alteration of the gene expression will affect the survival of many cells. Uh, then this gene cannot be used as a drug target. You really want to target those genes that have very weak or no correlation between expression and dependency. And a note here is that sometimes you may not find the genes you get as the top, negatively select genes on the website, but just try using its alias. Just maybe search Google it or search in NCBI gene database to find the other names for the same gene. And in question 4, it's very easy. We want you to identify druggable genes based on public data set. And first, we give you a pan essential.txt file that contains genes that are essential for all cells. And you want to remove those genes from the negatively select genes you got in question 2 and then get the top genes by sorting FDR. And you should go to this Oasis genomics.org and select glioblastoma and input the top 10 genes, and it will tell you which gene is druggable. But when I tested the homework 6, it seems that it's possible we cannot open this website for some unknown reason. So I found another website for you to try this one. And the similar idea, you should go to that website and input the genes. I think in this case you cannot select glioblastoma, but in any way, just input the gene and see if it will show any druggable information. So for question I think question 4 is the end of part 4. Yeah.